Today we will cover about this amyotrophic lateral sclerosis pathological mechanism. So this kind of diseases usually occur in the adulthood, and when the symptoms are starts to occur or becomes observable, then the patients will not have much time, as there is no clear diagnosis or therapeutic approaches available to slow down these infections. And this kind of disease might also cause paralysis, as it will affect the neurals in Spain, neurals that connects the spinal cord and brains. So there are three different models that are uh, shown here in this figure. So we will start with this panel A, which shows that it hypothesized that this kind of disease might occur because of the mutations in this SOD1 gene. And these mutations in the SOD1 gene will affect the chaperones, that is heat shock protein 70, and that will cause this reactive oxygen species ROS formations. And this will cause the oxidative stress within the cytosols of the cells, and that will leads to the dysfunctioning of the different organelle functions such as abnormal autophagy activations. And this abnormal autophagy activations will affect the functions of the mitochondria, and this kind of changes might observe in the neurons that will cause the neurodegenerations. And since we know that from our various previous videos that this oxidative stress will affect the organelle functioning of the Golgi apparatus and endoplasmic reticulum, and these two organelles plays an important role in the post-translation modifications. For instance, this endoplasmic reticulum plays a role for the initiations of the end-linked glycosylation, and the Golgi apparatus helps in the maturation of this end-linked glycosylation, and also the O-linked glycosylations occurs in the Golgi apparatus. So this will also get disrupted because of the oxidative stress, and all this will leads to the death of the neurons. So, and in the other models, so because and these things have been validated through a gene that is found in the mitochondria, and if that gene is get disrupted, then it will also replicate the similar kind of functions in the animals. So that genes could be the target point for the patients who are affected with this amyotrophic lateral sclerosis. So now let us go through this model 2. This model 2, what will happen is that this kind of disease will occur not only because of this SOD1 mutations, but also the mutations in the SOD1, the specific mutations in the SOD1 is this C9. Not only let me restart about this panel B. The panel B, uh, the problems occurs not only because of the SOD1 mutations, but also in the mutations that they have described as C9 or F72, and these mutations are in different gene. So I'm not able to recall that gene name, but you can go through the reference to know about more information about this mutant. So what will happen because of this mutant, and this mutant will lead to the formation of dipeptide that will interact with this affecting, and thereby leads to the increase in the amount of the calcium within the cells, and those might cause these affectins polymerizations formations and in the cytosols that can be seen here. And at the same time, these upregulations, this misfolding of this SOD1 proteins will cause this upregulation of this ROC and ROA pathways. These ROC ROA pathways will also promote this affectins rod-like structures formations that we have discussed, understand these relations in our previous videos also. So, and this will cause the decrease the cell motility, and ultimately leads to the death of the neurons. So now we will understand this last model, and it discusses also, it also considers the abnormal polymerization of these affectins that will cause or that will affect different functions within the cells. But the main main focus of this model is this disruption of this nuclear cytoplasm transport membrane, and this will occurs because of the abnormal formations, polymerizations of these effectins, and this disruption in this NCT, it is nuclear cytoplasm transport pore, will cause the abnormal functioning of these motor neurons, and also these effectins polymerizations will further increase and the myosins will decrease. And apart from this, the motor there is a mutations in the motor neuron that will increase the neurotoxicity within the cells. And apart from this, the WNT pathway will get hyperactive, and that will cause leads to the more beta actin formations. These beta actin formations will go into the cytosols, will go into the nucleus, and will cause the epigenetic transcriptional changes or the epigenetic changes, such as the downregulation of this tubulin alpha 4a, and this will promote, or this will disrupt the cell functioning, or this will aggravate the cell functioning which is already been disrupted. And also along with that, the mutant PFN1 will cause this abnormal autophagy, and that will also promote these neurodegenerations, and the actin assembly will be disrupted by this mutant PFN1 also. So all of these are related to this NCT dysfunctional NCT defects. This transport nuclear cytoplasm transport pore defects. So this is the third model. In this case, the NCT can be targeted in order to reverse this disease or treat or diagnose this disease. So these are three different models that are proposed for this uh, amyotrophic lateral sclerosis, and this could be the targeting point for the therapeutic development in order to diagnose the patients who are affected with this disease. If you need to understand more about this amyotrophic lateral sclerosis pathological mechanisms in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.